Good afternoon. I'm Professor Jana Asselman, and I would like to introduce you to our research, Innovations in Blue Life Sciences. In our lab, we focus on the interactions between oceans and human health, both positive and negative. Positive interactions are the health benefits from seafood or the ecosystem services such as primary production that we get from our ocean, but also marine biodiscovery, new drugs, biofuels, or industrial products from the ocean, or our health promotion through what we call the blue gym effect, the fact that we work out more when we're at the sea. But there are also negative effects for human health, such as pollution in our ocean, microplastics, chemicals, other litter, biological hazards, harmful algal blooms, parasites, bacteria, viruses, and even invasive species. And there is, of course, climate change. Ocean warming, ocean acidification, and sea level rise can all contribute to negative health effects. Now, we do have to note that all of these negative interactions between oceans and our own health are propagated in part due to our own impact on the ocean. Today, I would like to talk to you about our research in biodiscovery and plastic pollution. Marine biodiscovery, leveraging the ocean's biodiversity for new product applications, either from an industrial point of view or a human health point of view. We particularly focus on biomolecules in sea spray aerosols. Biomolecules or biogenic molecules, referring to the collection of molecules in the ocean produced by marine organisms. Biogenic molecules are present in the ocean, but we know that they tend to enrich in what we call the sea surface microlayer, the tiny layer of about a couple of micrometers on top of the sea surface. And it is when the molecules are enriched in these layers that they can end up in what we call sea spray or in aerosols. They can aerosolize through the formation of jet drops, film droplets, or spume drops. All of these occurring, of course, through the breaking of waves in our ocean. Now, why are sea spray aerosols so interesting? They are so interesting because the concentration of biomolecules can be 100 to 500 times higher than in the ocean. We have specifically shown this for phycotoxins. Phycotoxins produced by marine algae or diatoms end up in the ocean. However, through the enrichment in the sea surface microlayer, they eventually end up in sea spray aerosols. And we were able to quantify that their concentration in the sea and in the air. And we were able to show that they were enriched with a factor of 100 to 500 compared to in the ocean. This, of course, makes biomolecules in sea spray much more easier to detect than in the ocean. Now, how do we do this? We use a very simple aerosol sampler collected to a filter holder, and we basically collect air and molecules on that filter holder when placing them right near the seashore. We then take these filter holders to the lab and extract the biological molecules. These biological molecules are then end up in 96 well plates, where we expose them or where we expose human cell lines to these molecules to study human health effects. Using RNA sequencing, we were able to understand how biomolecules in sea spray aerosols and more particularly phycotoxin can affect human health and can be a source of novel drugs. We saw that sea spray aerosols affect the mTOR pathway. The mTOR pathway is a key signaling pathway in all our cells. Inhibition of mTOR is often associated with positive health effects, while stimulation of mTOR is often associated with negative health effects, diseases, and cancer. Using RNA sequencing, we saw that sea spray aerosols not only inhibited the mTOR pathway, but triggered a cascade of signaling pathways and networks, including interactions with the steroid biosynthesis, glycogenesis, spliceosome, and lysosome. We also saw the inhibition of PCSK9. PCSK9 is a major pharmaceutical target in both cancer research as well as cholesterol research. Basically, our results show that marine aerosols, and sea spray aerosols in particular, can be a very good source of biodiscovery. Our lab specializes in the collection and characterization of sea spray aerosols. We use both collections in the field as well as we have our own what we call marine aerosol reference tank to generate sea spray aerosols in the lab. And we of course also then study the human health effects using human cell lines and molecular methods. The next topic I would like to talk to you today about is plastic. We all know that the ocean is a source of plastic pollution. Plastic is often referred to as the larger plastics, but also the microplastics or the small plastics smaller than 5 millimeters. People often refer to microplastics as plastics between 1 micrometer and 5 millimeters, but we also focus on the larger and the smaller ones, particularly the ones in the nanometer range, as these can be most harmful to human health. Microplastic particles end up in the ocean through the degradation of large plastics, but also through wastewater from households and direct emissions. With our group, we collected water and sediment samples at different sites around the Belgian coast in the Belgian part of the North Sea, both at sea and on the beach. We also collected aquaculture organisms, seafood basically, both in the wild and from aquaculture farms. Using digestion methods, we characterized the plastic concentrations in all of these samples, focusing specifically on microplastics. We observed that microplastics were present in all the organisms that we collected, both in the mussels in the field, but also mussels from aquaculture, oysters from aquaculture, and a marine worm living in the beach sediment. All of them had different concentrations of microplastics in their tissue. Here you can see visual images of some types of plastics we found in these organisms. You can see that they differ in shape, size, and color, which is an indication of the wide diversity of plastic pollution present in the ocean. Now, the key part is, of course, where do these plastics end up in marine organisms? To get a better view on where plastics end up in seafood, we exposed marine mussels in the lab to fluorescent microparticles and then visualized the uptake using X-ray tomography at the Center for X-ray Tomography. Here you can see what happened to these plastics once they were taken up by the mussels. Using X-rays, we were able to look past the mussel shell and really go into the mussel tissue. As you can see, we're slowly using X-rays to remove the shell from the mussel and then are going into the tissues. Still no plastics, but when we go and specifically focus on the digestive gland, we see several things appear. We see our small green fluorescent microplastics that are taken up by the digestive gland. These larger dots indicate that these plastic particles also aggregate and form larger clusters in the mussels. Overall, with this study, we were able to show that mussels take up microplastics, which may of course have implications for human health. And that brings us to our next question. What does it mean for us if we eat up organisms contaminated with microplastic? To this end, we used human gut cell lines, CACO2 cell lines, and exposed them using fluorescent microplastics at concentration levels found in food sources. And we saw that these plastics could also be taken up by the cells. Here you can see the cells in red and the nucleus in blue. And you could see that the microplastics are on the outside, inside, and even moving to the nucleus inside the cells. Now the question is, of course, these are all in vitro results. Is there a risk? And that, of course, depends on the concentration. Anything can be harmful if only the concentration is high enough. Together with our collaborators at FLIS, we determined the concentration levels of microplastics in the beach, floating in the ocean, and in the seabed. We determined that currently, for ecosystem health, these concentrations are safe. But using projections based on plastic production, we saw that 100 years from now, there would be a significant risk for organisms living in the beach as well as the seabed. Now, for human health, there is still insufficient data to properly assess what levels of microplastic contamination or nanoplastic contamination would be harmful to our health, so we were not able to make a risk assessment for human health. To sum up, in terms of marine pollution and plastic expertise, our lab has experience in hazard assessment, the collection and the characterization of micro and nanoplastics in the field with a particular focus on marine environments as well as organisms and biological samples. We also have expertise in effects assessment, both at the environment and ecosystem level, using key indicator organisms such as mussels, but also with regards to human health, using in vitro studies such as human cell lines. We combine the results of both hazard and effect assessment to make risk assessments that are relevant for policymakers. And with that, I would like to thank you for your attention.